Mutations are changes in our genetic code. If you want a definition, like a Webster's dictionary definition, mutation is a permanent change in the nucleotide sequence of a genome that can be passed to future generations. We have two types of genes that we basically categorize. Wild type. It's a microbe with the naturally occurring non-mutated characteristics. The genome is the way that it has always been, no change. A mutant strain is a microbe with an altered or mutated characteristic. So their genome has been changed. It's not what it originally was. What those changes are, how they're altered, it all depends on what was changed. But the thing to take away is the mutant strain doesn't have the same genome as the wild type strain does. There we go. What can cause a mutation? Well, they can be induced or they can be spontaneous. Spontaneous is a literal random change in the DNA from errors in replication. So I talked about the DNA polymerase going back and proofreading. Sometimes it doesn't do a real good job. When it doesn't do a good job, you have changes to the DNA that weren't originally there. And it's spontaneous. Sometimes it just happens. However, you can actually induce mutations, an induced event. Mutagens are chemicals that we know alter DNA. You can get exposure to those. Mutagens are things like radiation, some chemicals. We often call them carcinogens because they're messing with your DNA and they can actually induce cancer cell formation. They disrupt the DNA in some way, shape, or form. Radiation can damage the DNA, and it does this by causing things called thymine dimers. Remember, thymine is the T in adenine thymine. If you have a thymine next to a thymine, it'll cause them to glue themselves together. And it means that you can't get them apart. So your correction machinery will actually go and take that chunk out and glue everything back together, and that messes up everything. Imagine a sentence and taking two letters out and then putting everything back together and hoping it makes sense. It's not necessarily the best idea. Some mutations are harmful. We call these lethal mutations. These are, as the name suggests, mutations that lead to the death of the organism. Some can be helpful. Some change some characteristic that allow the organism to have a benefit in their environment. Now, what that benefit is depends on what the mutation is, but the fact that they can be beneficial is also a good thing. Others are neither. Remember, we talked about wobble and how the third letter of a lot of the amino acids codons can be any of the letters and it's still the same amino acid. Technically, that is still a mutation. That still changed the genetic code. But because it didn't change what was in that position, which amino acid was in that position, it's technically a neutral mutation because it didn't change anything for the better. It didn't change anything for the worse. It just changed the code. Point mutations. Point mutations are small mutations that only affect one or two bases on a gene. There are several types of point mutations. There's a missense mutation. It's a change in a base that results in a different amino acid. This can be major or it can be minor to the cell depending on where it happens, what the change is. If the mutation is major, you get a non-functional protein. I need lactase to digest lactose. And I have this point mutation that causes it to misform. Imagine having a car with no engine. Will the car run? Obviously not. If you misfold a protein, it's like having a car with no engine. It doesn't work anymore because it's not oriented correctly. And with one amino acid change, you can completely destroy the structure of a protein. So if I have this major issue with the protein not working, this can basically be bad news for a cell. I have an enzyme that I need so that I can live, and now I can't make it anymore. It's going to be detrimental. If it's really minor, it might not affect the protein function at all, leaving the cell happy. Some amino acids are about the same size. They react to things the same way when they're folded. So I might be able to change things, one amino acid in a sequence, and it doesn't really bother anything. Remember I told you that you can have a chain of amino acids, but it has to fold. Some parts have to get cut out. Well, if I've got a chain of amino acids and that mutation happens in one of those sections that gets cut out anyway, it's not going to matter in the final function of the protein. So in that case, no effect at all, the cell's happy. Missense mutation. In lysine place, if it changed this middle base right here to a G, that would replace lysine with arginine. Both of these are about the same size. They're really, really similar in shape as far as chemically. With that in mind, probably with this mutation, even though I am changing lysine to arginine, this protein would probably still fold properly and would probably still work properly. This is an example of a mutation that happens. It changes the amino acid, but probably would be okay. However, if I change that middle letter of my code, nucleotide of my code, to a cytosine, that would put threonine in that spot of amino acids. The threonine is a completely different shape. Because of the different shape, when I try to fold this, it is not going to fold correctly, and I will end up with a non-functional protein. Depending on what that protein is, will pretty much determine whether or not this is going to be detrimental to my cell or something that I can live with because I didn't need that anyway. If it is something that I have to have, this is detrimental. This could kill my cell, literally, because my cell cannot function correctly anymore. It would be equivalent to taking the engine out of your car. Would your car run? No, it doesn't have an engine. If this is that type of protein that it has to be there so the cell can run, and I just messed it up, cell's a goner. A nonsense mutation. A nonsense mutation is a change in a base that results in a stop codon, causing the protein sequence to terminate earlier than was planned, according to the sequence. This almost always results in a non-functional protein because I literally didn't make the protein I was supposed to make. I might have an amino acid sequence originally coded for that was 110 amino acids long, and at base 30 or at codon 38, I hit a stop. Well, if it was 110 amino acids long and now I'm stopping at number 38, that's not going to work anymore because I don't have all the parts that I need. If we go back to my car analogy, imagine putting together the body of the car, putting the engine in, and that's all we do. There's no windshield, there's no tires, there's no axle, there's no undercarriage. It wouldn't be a functional car because not all the pieces are included. A nonsense mutation. A nonsense mutation happens when there is a change in a base that results in a stop codon. I mentioned this, I believe, in the lecture where I said if we have 110 amino acids in a row that we need in order for it to be functional, and I end at 30 something, it's not going to work. In my original wild type, I have 1, 2, 3, 4, 5, 6, 7, 8, 9 amino acids. If you change one base in cysteine, that last one to an A, this becomes a stop codon. Thus, it stops with methionine, serine, and tyrosine. Cysteine isn't there, lysine isn't there, glycine isn't there, nothing else is there. It literally stops right here. Non-functional protein, I didn't finish it. I don't know how many of you have seen Elf. In Elf, Buddy's father puts out a book that doesn't have an ending. The last, I think, 7 pages are missing. If you don't have the end of the book, you don't know what happens. If you don't have the middle of the book, you don't know what happens. And in this case, because I stopped it early, you don't have a complete piece anymore. You've chopped it off before it was finished. A silent mutation. This is a change in a base that does not change the amino acid. It does not produce a change in the cell at all. Wobble. I can change that third base on several amino acids in the list that I showed you, and it doesn't change which amino acid is there. Did it change the sequence? Yes. So is it a mutation? Yes. But ultimately, did it affect anything? No. I still have the same amino acid in the spot I need it, so everything should work fine. Silent mutation. Silent mutation, you are changing one of the bases, one of the nitrogenous bases, but it remains the same amino acid. That effect of wobble comes into play here. Cysteine, UGU in the original wild type sequence. If I change it to UGC, it's still cysteine. It's not changing the sequence of amino acids at all. Did it change the sequence? Yes. The base sequence, yes. The genotype, yes. What does it say? It now says something a little bit different, but is the meaning the same? Yes, I still get a cysteine in that spot. So does it read the same? Yes. So it's a silent mutation because the actual sequence of amino acids didn't change. The sequence of bases did, but not the amino acids. So my final product is still my final product. My final sequence of amino acids is the exact same thing, regardless of this change in the nitrogenous base sequence. Back mutation. It's a change in a base where the mutant gene, the thing that changed, reverts back to the original version, to the wild type. Does it happen in real life? Yes. Why? I don't know. But sometimes a mutation will just kind of reverse itself. When it does, when it's something that could potentially be dangerous, this is a very good thing. When it does, when it's a silent mutation, it's good that it did, but it didn't really change anything. Back mutation. I revert back to what I originally was. This was my silent mutation where I changed the third base. It still stayed cysteine, but the actual sequence of nitrogenous bases was different. Amino acids stayed the same, but that sequence changed. If I were to take that C and replace it with a U, the original wild type sequence, that would be a back mutation. I believe I said this in the other lecture. Why does it happen? But sometimes it does, and when it does, we just call it a back mutation because it started looking like this, it ended looking like that, which was back to wild type. Frameshift. Frameshift is a deletion of one or two bases, which results in a changed reading frame, which alters how the codons are read during translation. This is going to result in a non-functional protein. Almost always, this is very detrimental. And it's very detrimental because I change the protein from wherever this deletion happened all the way to the end. Even my stop sign won't necessarily be a stop sign anymore. Frameshift mutation. A deletion of 1 or 2 bases, which results in a changed reading frame, which alters how the codons are read during translation. Remember that every 3 letters is a specific code for a specific amino acid. In a frame shift, I delete 1 and everything kind of shifts the other way. So in this case, let's say I delete that. All of these letters shift this way. Now I added a few after my stop because normally there are actually a few more codons after, but since we hit the stop, it stops and it doesn't keep going. But in this case, because of this shift, it's going to change how this entire thing reads. So methionine would still be there. Serine would still be there. AUG, UCU. However, this would start reading every 3 letters like this. If you look, there are still 3 letters in between each of those red lines, but it is completely different than what we had. So UAU now becomes UUU. UGU now is GUA. AAG is AGG. GGU is GUA. UUA, GUG, UCU, 3 A's, GUA, GAC, and then we have these 2 C's hanging out here that won't be anything. If you look at this sequence compared to what was up there, it is now completely different. What is being coded for as far as the amino acids go, also completely different. This is still methionine. This is still serine. This is no longer tyrosine. Instead, this is phenylalanine. No longer cysteine, instead it's valine. Valine. No longer lysine, it's arginine. No longer glycine, this is valine again. No longer asparagine, it's leucine. No longer serine, it's again valine. No longer valine, even though we have 3 valines. Now I've got serine. The AAA is lysine. The GUA is, again, valine. And the GAC is aspartic acid. and we literally have no stop anymore. Frameshift is almost always going to be a detrimental mutation. It completely changes what this says to put together. The recipe changed, and I didn't even take 2 of my bases out. I took 1, and look at the mess that I now have compared to what I started with. Frameshift, I've never heard of Frameshift not being detrimental, to be honest. I don't know everything, but the amount of change this causes simply from shifting that reading frame, you end up with something completely different that is nothing like what you originally planned for. The repair of mutations. DNA has a proofreading mechanism to repair replication mistakes. This is that I told you about, that it goes back and there's a dip, there's a bump, it goes, wait a minute, what? The cell also has UV damage repair systems to work on a small number of UV mutations. So going out in the sun for, an hour, maybe once a month, we do have mechanisms in our body that are designed to try and repair those things. It can also use excision repair to remove defective bases. See how these do not match? This is an oval, this is a triangle, and these wouldn't fit together. Enzyme 1 comes in and cuts out the piece that is mismatched, the piece that isn't supposed to be there. When it does that, another enzyme brings in the proper nitrogenous bases, the proper nucleic acids, and fuses them into place, repairing the DNA. Will this work for everything? No, it can't. There is not a universal repair mechanism like that. Will it work for small things? Yes. But if you were at Chernobyl in the 80s and got massive doses of radiation, the amount of damage to your DNA would be so extensive, little things like this wouldn't work. Push comes to shove, we can repair small things. Big, massive damage remains big, massive damage. There's nothing that can fix that.